Hello, my name is Mandisa Holmes, and I'm here to talk to you all about crystallography. Crystallography refers to the field in which studies crystalline structures, such as gemstones and rocks. In this field that we study, crystallography is used to study proteins as well as other molecules. Now, you might be saying to yourself, crystallography, that sounds familiar. That's simply because we did discuss this in Module 4's Protein Expression and Analysis, in which we discussed how crystallography is used to determine what the actual structure of the protein looks like. But with this presentation, I aim to dig a little deeper and further that explanation. X-ray crystallography has been fundamental in the science field for many years now, and is the technique that is used. X-ray crystallography has been fundamental in the science field for many years now, and is the technique that is used to determine high-resolution 3D crystal structures of either atoms or molecules. In the early years of X-ray crystallography use, it was used in determining the size of atoms, the atomic scale difference between various materials, the lengths and types of chemical bonds, grain orientations, and many other things. But in today's world, it's mostly used to determine the size of atoms. In this realm of science, we use it to reveal the structure and the functions of molecules, such as drugs, vitamins, proteins, and nucleic acids, being used mostly in determining how pharmaceutical drugs interact with its target proteins. Now let's get into the workflow of X-ray crystallization. There are 4 important steps to follow in order to determine the structure of protein by X-ray crystallography. These steps are crystallization of a protein, collection of a diffraction pattern, calculation of the electron density map, and production of an atomic model. The first step is seen to be as the hardest, simply because it's hard to produce an adequate crystal that is large enough with all dimensions being bigger than 0.1 millimeters, also being pure in composition and having no imperfections. The most widely used technique to achieve such is vapor diffusion. In vapor diffusion, a drop that contains a mix of the precipitant and protein solution is closed into a chamber containing a pure precipitant. The vapor from the water then begins to diffuse out the drop until the concentration of the solution of the drop as well as the precipitant are equal. The crystals are then placed into an intense beam of X-rays that usually are a single wavelength, and that produces a reflection pattern. Each compound produced has its own unique diffraction pattern in which the angles and the intensities of the diffracted X-rays are measured. From this diffraction pattern, an electron density map can be derived. The electron density map is a 3D plot that shows in what areas the electrons are concentrated within that crystal. Those areas that contain high density are seen to be the areas that contain the protein atoms, and from there an atomic model can be produced. A positive and negative control is the obvious answer here. Using a positive control, we can cross-compare while using a well-known specific protein. Using a negative control, we can use a crystal that will not create a diffraction and using it to compare the structure that we produced. Let's get into the limitations and advantages of crystallography. Some of the limitations include finding a sample that is actually crystallizable, producing an adequate crystal. If you all remember what we discussed earlier, the hardest part about crystallography is indeed producing an adequate crystal, simply because it must be large enough, lack any imperfections, be pure in composition, and all dimensions must be larger than 0.1 millimeters. Another limitation is the type of samples that can be analyzed. For example, larger molecules as well as membrane proteins are difficult to crystallize. Some of the advantages include the fact that there's no damages done to the samples, it's high resolution, high performance, and high precision, free of pollution, and low environmental requirements are needed. This figure is one of which that I borrowed from the X-ray crystallography manuscript. If you look closely, you can tell that this figure is a picture of a crystal of a bovine piconavirus, which measures at 0.2 milliliters. which is actually the longest dimension grown using ammonia sulfate as the precipitate and using microdialysis as the apparatus. Now, if you remember from earlier in the lecture, anything greater than 0.1 milliliter, as far as dimension go, is a golden crystal. So this crystal of the bovine piconavirus is a star in itself. The crystals here took about 48 hours to grow at room temperature and are very fragile simply due to the virus particles in the crystal being spherical and 300 angstrom in diameter, which in turn makes the solvent very high and the particle contacts very low. Please note that this crystal is a result of vapor diffusion. This figure is one from a manuscript called Protein Production in E. coli for Structural Studies by X-ray Crystallography. This table represents diffracting crystals and structural data from M. tuberculosis and P. aeriform proteins. This manuscript goes to explain that E. coli remains the most popular for heterologous expression of recombinant proteins. It goes on to list the advantages and wraps it up by saying that E. coli is the host choice for metabolic labeling with Salaminthin 09 for the multi-wave anomalous diffraction, phasing in the X-ray crystallography. This table further explains that with the protomes P. aeriform and M. tuberculosis, the majority of the proteins are crystallized and are expressed in E. coli. This last structure is from a manuscript entitled Structure Determination of GPCRs, Cryo-EM compared with X-ray crystallography. In this article, it takes a look at G protein coupled receptor structure and compares the structures that were created from X-ray crystallography and Cryo-EM. Up until recently, X-ray crystallography was the only determination of the structure of the GPCR. Figure 1E simply shows the cumulative number of the G protein coupled receptor structures that were determined by the X-ray crystallography and those that were determined by the Cryo-EM. In this chart, you can see that both include multiple structures of the same receptor that are bound to different ligands, different species, and also different intracellular binding partners. Thank you all so much for listening, and I hope you learned something and enjoyed my presentation.